Today we will be talking about regulation of gene expression, meaning a specific gene, if it is getting expressed, how is it regulated? And in that we will be talking about the operon. Now operon is something that I've already spoken to you about when we were talking about different structures within the DNA, wherein you have things like a cistron or a recon or a muton or things like that. When I was talking about that, I have already touched on operon, and here I'll be briefly talking about it again. An operon is basically the functioning unit of a DNA. So within the entirety of the DNA or within the entirety of an organism's genome, if there is a functioning part within the DNA, that functioning unit, that functioning part is known as the operon. So operon is basically the functioning unit within the structure of the DNA. Now operon basically has genes. It can be a single gene or a couple of genes, all of which are controlled by one single promoter. Okay, so a single gene or a couple of genes, more than one gene, which are all controlled by one single promoter, and that entire structure is known as an operon. So multiple genes which are controlled by one single promoter. What is a promoter? Promoter is something that we have already talked about before. It's that part of the DNA which is a part of an operon, and it's a part of the DNA where things like transcription factors will come and join or attach to the DNA strand and help in transcribing the gene itself. So the gene needs to be transcribed and give rise to a mRNA, and this mRNA will get translated to give rise to a protein. So for the transcription to occur, for it to get initiated, you need the transcription factors to come and attach to the promoter region in the DNA, and then RNA polymerase can come and attach, and then the initiation of transcription will take place, and then the gene will get transcribed, which will give rise to a mRNA. The mRNA is going to give rise to a protein. So this is something we have already touched on already. So going on with the operator, the operon concept. Now, the operon can give rise to a polycistronic mRNA. So like I was saying, it can be one single gene which is controlled by one promoter, or it can be multiple genes which are controlled by one promoter. Okay? So if it is multiple genes which are controlled by one single promoter, then you are going to have a polycistronic mRNA. Okay, meaning that one single promoter is controlling the expression of 2 or 3 genes, meaning that you will have a mRNA which will have the translational part for 3 genes. So it's going to give rise to 3 different functional proteins or 2 different functional proteins, and this is something that is known as a polycistronic mRNA. Now polycistronic mRNAs are specifically found in prokaryotes. Prokaryotes are the ones that usually have polycistronic mRNA, mainly because they do not have introns and exons. So there is no differentiation between introns and exons when it comes to prokaryotes. That is something that is found only in eukaryotes, and therefore eukaryotic mRNA needs to undergo post-transcriptional modification, whereas prokaryotic does not undergo post-transcriptional modification, and that's why in prokaryotes you have polycistronic mRNA. In eukaryotes you have monocistronic mRNA, meaning that one promoter is going to control only one gene. The expression of one gene is going to be controlled by one promoter. Alright. Now when you think about polycistronic mRNA, the polycistronic mRNA can undergo something known as trans-splicing. Okay. This is a phenomenon wherein polycistronic mRNA, which has been transcribed, will have the mRNA will have the genes for 3 different proteins, right? But after the transcription has occurred, this mRNA is going to get chopped off into 3 different gene portions. Okay, so this polycistronic mRNA is going to get chopped off, and it's going to give 3 different monocistronic mRNAs, which are going to be translated separately by ribosomes to give 3 different proteins. So that is a phenomena that can occur in case of prokaryotes, but usually it's a polycystonic mRNA which is going to be translated together, giving rise to 3 different proteins. But transsplicing can also occur. Okay. Now the operon that we will be talking about here specifically would be the lac operon. Lac operon is the first operon that was discovered by Francois Jacob and Jacques Monod in the year 1961, and they got a Nobel Prize for their discovery in 1965. Since it's the first operon that was discovered and also the operon that is most studied, that is the operon that we will be going to be studying today. Now, why did they come up with the idea of operon? They came up with the idea of operon because they realized that a cell cannot be synthesizing all of the proteins all the time. So, the cell is going to synthesize a specific protein only when it requires that protein. So, proteins basically act as enzymes. So, every enzyme that you see, majority of the enzymes at least, because there are RNAs also which act as enzymes, but we are not going to consider that for now. We are going to be only thinking about proteins. So, majority of the enzymes are all proteins. So, they all give rise to some sort of a catalytic activity. They are going to perform a specific function. And if they are performing a specific function, they need to be synthesized or they need to be available only when that function needs to be done. Otherwise, they need not be there. So, Jacques Monod and Francois Jacob, they came up with the idea that a cell is going to synthesize a specific protein only when it needs it. If it does not need the protein, it is not going to synthesize it. But they wanted to figure out how and when this synthesis occurs. So, what is controlling the synthesis? How does a specific protein get synthesized or the synthesis is stopped? What is controlling all of these things? And that is how they came up with the idea of the operon concept. Now, before we get into the lac operon as such, there are certain aspects of a operon concept that we will need to be talking about. First things first will be the promoter. Again, promoter is a thing that we have already studied, specifically when we were studying transcription. So, promoter will be a part of the DNA which is before the gene, so before the first codon, which will be AUG. Before that, there is a sequence of the DNA to which transcription factors are going to attach themselves, and then that is going to bring in the RNA polymerase and it is going to initiate transcription. So, promoter region is the part of the DNA where transcription factors are going to attach. They are going to bring in the RNA polymerase and they are going to start off the transcription process. So, that is something we have dealt with in detail specifically when we were studying transcription. The next part will be a operator. Now, this is a part of the DNA which is between the promoter and the first codon. So, if you have a part of the DNA that is between the promoter and the first codon, meaning the AUG codon, before that where the gene starts, that part of the DNA is known as the operator. Now, what is the function of the operator? The operator is going to be a region of the DNA where a repressor is going to attach itself. So, whenever a repressor is attached to the operator, then polymerase cannot do its action, which is to transcribe an mRNA, because when on the DNA strand a repressor is present, because operator is just a part of the DNA, and on top of it, if there is a repressor attached to it, it is a physical barrier for the movement of the RNA polymerase, and therefore the RNA polymerase cannot move physically on the DNA strand, and because of this the transcription process is halted. So, operator is the part of the DNA where a repressor comes and attaches itself. That brings us to a repressor. Repressors are usually protein molecules which are synthesized by their own genes. Okay? So, repressor has its own gene which is controlled by a promoter, and that promoter is always active. So, it does not need an induction, it is always active, it is always synthesizing the repressor, and the repressor is always a protein molecule which is going to come and attach itself to the operator region. Okay, and the repressor is going to attach itself to the operator region whenever there is no inducer present. So as long as the inducer is not present, the repressor is going to attach itself to the operator region. Now there are things known as co-repressors. Co-repressors in case of prokaryotes. So if you're thinking of prokaryotes, co-repressors are usually small molecules. In case of eukaryotes, they are always proteins. Okay, so co-repressors in case of prokaryotes are always small molecules. In case of eukaryotes, they are always proteins. What do co-repressors do? They are going to be attaching themselves to the repressor, and they are going to enhance the activity of the repressor. So co-repressors are those molecules which are going to attach themselves to the repressor and make the repressor attach itself to the operator. Got it? So co-repressors are helping the repressor in attaching itself to the operator region on the DNA. Okay, and in case of prokaryotes, they are always small molecules. In case of eukaryotes, they are always protein molecules. The next thing will be activators. Now activators are again protein molecules which attach themselves to the enhancer region. Okay, so they are going to attach themselves to the enhancer region, which is present either before the promoter region, right next to it, or further along from the promoter region. So enhancer, like the name suggests, is going to enhance the function of the promoter. Okay, promoter by itself is going to help the RNA polymerase to do transcription, but enhancer is going to help the promoter to do the work even more easily. Okay, so enhancers are going to specifically enhance the action of the promoter, and the enhancer gets activated whenever an activator is going to come and attach itself to it. And activators again are proteins, and they do the exact opposite work of a repressor. So repressor is going to stop the RNA polymerase from transcribing a specific gene. Activator is going to help the RNA polymerase in transcribing the gene. Okay, so these repressor and activators work opposite to each other. Alright, and activators are going to be attaching themselves to the enhancer region. The repressors are going to be attaching themselves to the operator region, and enhancers, when they are attached to activators, are going to help the promoter. Repressors, when they attach themselves to the operator, are going to stop the promoter, or rather the RNA polymerase, from transcribing the specific part of the gene. Okay, so these are the different regions within an operator that you will need to remember whenever we are going to be talking about a operon. Moving on with the lac operon itself, there are of course promoter and an operator region in the lac operon gene, and there will be three structural genes which are there in the lac operon. The first structural gene is the LAC-Z gene. So structural genes are what we would call as cistrons. These are the ones that are going to give rise to the mRNA, which is going to get translated into a protein. So structural genes are the ones that are the actual gene part, the actual functional part of the DNA that is going to give rise to a mRNA, which is going to give rise to a protein. So in case of the lac operon, it does have a promoter and an operator, and after the operator you have three structural genes. LAC-Z or the LAC-Z gene, which is going to give rise to the enzyme or the protein beta galactosidase, and its function is to break down lactose to one molecule of glucose and galactose. So lactose is a disaccharide, and when it is broken down, you are going to end up with one molecule of glucose and one molecule of galactose, and that's its function. Then you have LAC-Y gene, and LAC-Y is going to give rise to the protein beta galactoside permease, and this protein is an intrinsic protein, meaning that it is going to embed itself in the plasma membrane. So it is an intrinsic protein that is going to embed itself in the plasma membrane, and because it's a permease, its function is to allow lactose from the outside of the cell to enter into the cell. So it's a transport protein that is going to allow the lactose molecule, which is present in the outside of the cell, outside environment of the cell, to enter into the cell. So that is the function of LAC-YZ gene, and you have the LAC-A gene, which is going to give the protein beta-galactoside trans-acetylase. And because it's a trans-acetylase protein, it is going to take the acetyl part from acetyl-CoA. So from the molecule acetyl-CoA, it's going to take the acetyl part and then transfer it to another molecule. Now, what is the actual function of beta-galactoside transacetylase in the lac operon? It is not known. As of now, we don't know what is the function of beta-galactoside transacetylase, but because it's an acetylase, it's going to transfer the acetyl part from the acetyl-CoA and transfer it to another molecule, maybe galactose or glucose, but it is not yet known what is its function. And it is of course going to have a repressor molecule. The repressor molecule in the lac operon is termed as LAC-I, and it is controlled by a constitutive promoter. A constitutive promoter is a promoter that's going to be active all the time, or it's going to be on all the time, meaning that the repressor molecule is going to be synthesized continuously. Okay, so when you have a constitutive promoter, it means that it is active, or it is on 24/7. It is always on. It does not stop producing the repressor molecule. So this LAC-I gene is always going to get transcribed to an mRNA, and that mRNA is going to give rise to a protein, and that protein is the repressor protein. Okay, so these are the structure of the lac operon. You have the promoter, the operator, the structural genes LAC-Z, LAC-Y and LAC-A, and then you have a repressor gene LAC-I, which is going to be present before the promoter of the lac operon, and it is controlled by its own promoter, which is the constitutive promoter, meaning that it is always on. Of course, at the end of the structural gene, you will have the termination part, which is going to have a stop codon, and that's pretty much all of the different structures that are there within the lac operon. So, how does the lac operon play into actual growth of cells, or how does it work? And lac operon is something that is in prokaryotes, so we will be taking E. coli as an example in this case. So when E. coli is grown in glucose medium, which is its primary source of energy, bacteria are going to grow pretty fast. They are going to grow exponentially because glucose is readily available and they can use it as their energy source to take it up and then grow. They are going to grow and divide, and the number is going to grow exponentially. Now, at the same time, when E. coli are grown in glucose medium, because lactose is not there, the repressor, which is being synthesized continuously, is always going to attach itself to the operator region. And whenever the repressor is attached to the operator region, even though the RNA polymerase can come and attach itself to the promoter region, it cannot transcribe the rest of the genes. So the LAC-Z, LAC-Y and LAC-A genes are not going to get transcribed. There is not going to be an mRNA, and therefore you are not going to have any of the enzymes, the proteins, to be synthesized. Because of this, the level of enzymes like beta-galactosidase or permease or transacetylase, all of these three enzymes, the number of these enzymes is going to be very, very low. Not that it is not going to be there in the cell at all, just that it's going to be extremely low. Now, transfer the same E. coli to a growth medium which contains lactose. As soon as you transfer E. coli to a medium that contains lactose, the cells are going to stop growing for a certain period of time. There will be a short interval during which time the cells are not going to grow any longer, they are not going to divide, there is not going to be an increase in the cell number, and that is because the lac operon is going to get started at that point of time. Now remember, I was telling you, when the glucose medium is there and the cells are grown in glucose medium, the number of the enzymes beta galactosidase or permease or transacetylase is going to be low. Not that it is not going to be there in the cell at all, just that it is low. So when it is grown on lactose medium, the few number of permease proteins which are there are going to allow lactose to enter into the cell, and this lactose will act as a inducer molecule, or rather the inducer molecule is termed as allolactose. So you have the inducer molecule which is the allolactose, or you can also term it as just lactose. So this inducer molecule is going to attach itself to the repressor molecule, and whenever the inducer is going to attach itself to the repressor molecule is going to undergo a conformational change. So when we were talking about protein maturation, I was telling you that there is a primary structure, a secondary structure, a tertiary structure, and a quaternary structure, and these structures need to be in a specific shape for the protein to do its function. So whenever the inducer attaches itself to the repressor, that conformation, that three-dimensional shape that the protein has is going to get changed, and because of this reason the repressor cannot attach itself to the operator anymore, and now there is no physical barrier sitting on the template DNA, because of which reason the RNA polymerase can easily transcribe all of the three genes which are there, LAC-Z, LAC-Y and LAC-A, and then you are going to get a polycistronic mRNA which is going to get translated, and you are going to have all of the enzymes present like beta galactosidase, permease and transacetylase. So within this short interval of time, when the bacteria are not growing any longer, as soon as they have shifted onto the lactose medium, there is going to be 1,000-fold increase in the enzymes. And as soon as the enzymes are going to increase in number, they can start using lactose as their energy source. So first the permease is going to allow lactose to come inside, and then beta galactosidase is going to break down the lactose molecule to one molecule of glucose and one molecule of galactose, which can be used as an energy source by the cell itself to grow and divide, which is going to occur afterwards. But there will be a short interval of time when the cells are not going to grow initially, when they are being shifted onto a lactose containing medium. Now what happens when lactose is removed from the medium? As soon as the lactose is removed from the medium, there is no more inducer. So this inducer molecule, which was the lactose, which was binding to the repressor and not allowing it to bind to the operator, is no more there. And because of this reason, the repressor is free to bind to the operator, and then the RNA polymerase cannot transcribe, so the lac operon is again not functioning, or what we call as lac operon being off. Okay, so whenever the repressor is not attaching itself to the operator and the RNA polymerase is transcribing all of the genes that are there in the lac operon, then that operon is known as an operon that is on. And when the repressor is going to attach itself and there is no transcription occurring, then it is known or termed as the lac operon being off, or an operon being off. So when there is no transcription occurring, the operon is off. When the transcription is occurring, the operon is on. And this is how the lac operon functions. Okay. The next thing that we will be talking about, or an important thing that you need to remember, is inducible and constitutive genes, or inducible and constitutive promoters. Now constitutive promoter is something that I've already talked about. So these are promoters that are going to be controlling things like the repressor, which are continuously active. So this promoter does not require any inducer. It does not require any sort of help for it to transcribe the gene that it controls. So Whenever a constitutive promoter is there, then that gene is going to get transcribed all the time, and it's going to get translated all the time, so that protein will be available all the time. As opposed to a inducible promoter, an inducible promoter will be something like the lac operon, wherein you need the inducer to attach to the repressor, and the repressor is not going to attach to the operator, and only then the promoter can function, meaning that the genes are getting transcribed. So an inducible promoter is something that is controlled by something, and it can be switched on and switched off. But a constitutive promoter or a constitutive gene is something that is continuously being synthesized, and it cannot be switched on or switched off. So those are the only two differences between a inducible and a constitutive gene, or an inducible or a constitutive promoter. We'll be talking about different kinds of operons that are present. So there is negative operons and positive operons, and there are two different kinds of negative operons and two different kinds of positive operons, and that we will be discussing here. First off, with negative operons, you have two kinds of negative operons: negative inducible operons and negative repressible operons. Now, negative inducible operon, an example would be the lac operon, wherein the repressor is always bound to the operator. Okay, so the repressor is always being synthesized and it is always bound to the operator unless the inducer is present. Okay, in case of the lac operon, the inducer will be lactose. So whenever the inducer is present, that is lactose is present, it is going to bind to the repressor and it is going to stop the repressor from binding to the operator, then the transcription will take place. So negative inducible operon, repressor is always bound to the operator unless and until the inducer is present. So whenever the inducer is present, the repressor is not bound to the operator. If the inducer is not present, the repressor is always bound to the operator. Okay, moving to the next one, negative repressible operon. An example for this will be the trip operon, which is the tryptophan operon. So this is the tryptophan operon, which is known as a trip operon. Now in this case, the repressor is always not bound to the operator. Okay, the repressor is always not bound to the operator unless and until a co-repressor is present. Okay, so the repressor is always not bound to the operator, but whenever the co-repressor is present, and this co-repressor is going to bind to the repressor and it's going to help the repressor in binding to the operator. So remember what I was talking before, that co-repressors are going to help the repressor in binding to the operator. So in case of negative repressible operon, the repressor is always not bound. It is not bound. In normal cases, it is not bound to the operator. Only when the co-repressor is present, and when co-repressors bind to the repressor, then the repressor can bind to the operator and the transcription is being stopped. Okay, so in this case, the operon is always off, and it becomes on only when the inducer is present. So whenever the inducer is present, the operon is on, but it is always off. Always the operon is off. Only when the inducer is present, the operon is on. In case of the repressible operon, the operon is always on. So its natural state is that it is always on, but whenever the co-repressor is present, the operon is off. Okay, so they are like opposites. Got it? So negative inducible operon is always off unless and until the inducer is present, then it is on. Negative repressible operon, it is always on unless and until the co-repressor is present, then it is off. On to the positive operons. Positive operons are also of two kinds. You have positive inducible operon and positive repressible operon. Now, in case of negative operons, the protein that was being used is the repressor. In case of the positive operons, the protein that is being used is the activator protein. Now, in case of positive inducible operon, the activator is not bound to the enhancer region. So, the activator is not always, in the normal condition, it is not always bound to the enhancer region unless and until an inducer is present. So, whenever an inducer is present, it is going to bind itself to the activator, it is going to change its conformation, then this activator can bind to the enhancer region, which is going to help the promoter region, and then the transcription is going to take place. Okay, so whenever an inducer is present, only then the activator can bind to the enhancer, which is going to help the promoter, and therefore RNA transcription can occur. But normally, under normal conditions, the activator is not bound to the enhancer. For positive repressible operon, the activator is always bound to the enhancer. Okay, for positive repressible operon, the activator protein is always bound to the enhancer region of the DNA unless and until a inhibitor is present. So, whenever an inhibitor is present, this inhibitor is going to bind to the activator, which is going to change its conformation. So, this cannot anymore bind to the enhancer, and therefore this is not going to help the promoter, and therefore transcription is not going to take place. Okay, so thinking about whether or not the operon is off or not. Now, because the activator is not bound to the enhancer, the operon normally is off unless and until a inducer is present. So, whenever an inducer is present, then the operon is on. So, normally positive inducible operons are off unless and until a inducer is present. Whenever an inducer is present, then the operon is turned on. In case of positive repressible operon, because the activator is always bound to the enhancer, normally the operon is on. But whenever an inhibitor is present, the operon is being switched off. Okay, like in the negative operon, in positive operons are also, they are also opposites. So, inducible operon is always off, and only when the inducer is present it is turned on. Repressible operon is always on. Whenever the inhibitor is present, it is turned off. In case of negative operons, the protein that is being used is the repressor protein. In case of positive operon, the protein that is being used is the activator protein. And the region where the repressor joins is the operator region, and the region where the activator joins is the enhancer region. So, these are the differences between the positive operons and the negative operons, and the individual positive and negative operons. Human Genome Project. What is Human Genome Project? Why did we do it? What is the reason behind it? And what is the uses of it is what we will be discussing throughout this topic. Now, Human Genome Project is basically the sequencing of the entirety of the genome and identifying and mapping all of the genes that are there in the human genome, both from a physical and a functional standpoint. Now, let's break that down. First things first, it's the sequencing of the human genome. So, the Human Genome Project was when the entirety of the human genome was sequenced, meaning that the sequence of the nucleotides that are there in the human genome was figured out. So, genes, DNA, the genome is made up of 4 nucleotides. You have adenine, guanine, cytosine and thymine. Now, the way that they are arranged one next to each other is the sequence of the DNA, and the entirety of the sequence of the human genome, which is close to 3 billion bases that are there, 3 billion base pairs that are there in the human genome, was sequenced. So, the exact way in which these nucleotides are arranged, so A, C, C, T, A, G, C, so like this, the 4 nucleotides are arranged in a specific order in the human genome, and that was figured out through sequencing. of the human genome. The next thing that they did was to identify and map all genes. So identifying and mapping all of the gene both from a physical and functional standpoint, meaning that they identified from the physical standpoint where the gene is present. So physically, in the entirety of the human genome, where a specific gene is present, it's present from 3,262 base pairs to 6,346 base pairs. So a specific gene is present at a specific location between these nucleotides. So that is identifying where it is present. Mapping is something that we have already talked about when we were talking about linkage in Mendelian genetics, is on the chromosome where a specific gene occurs. So that is mapping, and that was also again the physical aspect from the physical standpoint. So identifying where the gene is present in the DNA sequence on the genome, so that is present between 3,000 something base pairs to 6,000 something base pairs, is gene 1. And then another gene is present from 3,200,000 something base pairs to 600 something base pairs. Okay, so the physical location on the DNA, and also mapping wise, meaning that where it is present on the chromosome and what is its connection between another gene, which comes for recombination. So that was also figured out. And the functional standpoint is wherein we identified what is the function of that specific gene. So we know that a specific gene is present from 3,000 something base pairs to 6,000 something base pairs. But what is the function of that gene? What is the protein that it is giving rise to, and what is the function of that protein? That is again from the functional standpoint that was figured out. Also mapping from the functional standpoint, meaning that how recombination is going to occur. So how is it going to get passed on from one generation to another generation? How is it going to affect a population? How is it going to affect a specific trait from being seen or not being seen? How is it going to affect evolution also to a certain extent? So all of that was figured out from the Human Genome Project. It was a huge project that took 15 years in the making. The project itself started from 1990 to 2000, but the idea was pitched in 1985, and there were countries that took part in it was the United States, United Kingdom, Japan, France, Germany, Canada and China, and close to 20 different universities and research institutes within these countries took part in it. The funding for all of these experiments that were conducted majorly came from the National Institute of Health, or NIH. So NIH is the National Institute of Health, which is present in US. So the major funding agency was NIH from US, but there were also other funding agencies from different countries which funded this project. Now, at the entirety of the project, there were certain methodologies that were used. So, when sequencing the human genome or identifying and mapping all of the genes that are there in the human genome from a physical or a functional standpoint, there were certain methods that were used. And the methods that were used were these 4 ones which are left here. So the first method was to identify DNA sequences that gave rise to RNA, and this is easily done because within a cell you have certain genes which are being certain genes which are getting transcribed and then giving rise to an RNA, and it need not always be a mRNA. It can be a rRNA or a tRNA or siRNA, miRNA, any kind of RNA. So whenever transcription is happening, it's not always a messenger RNA or a mRNA that is being transcribed. It can be any kind of RNA. So if we take any kind of RNA that is there in the cell, any specific human cell that you take up, you can use that RNA to create a DNA molecule, and that is through the process of reverse transcription. And reverse transcription is something that I've already spoken about when we were talking about the central dogma of molecular biology. So when we're talking about the central dogma of molecular biology, reverse transcription is something that I've already touched on. Reverse transcription happens with the help of the enzyme reverse transcriptase. That is when using a RNA molecule, you can synthesize a DNA molecule. So using all of the RNA that is there in the cell, we can synthesize a DNA molecule, and figuring out where is the complementary sequence for that newly synthesized RNA, we can figure out which part of the DNA sequence is going to give rise to the RNA that we had. So you just take the RNA that is there in the cell, use reverse transcriptase, create a DNA molecule, figure out where this DNA molecule fits into the human genome completely. So where does it have its complementary base pairing, and then you have the DNA sequence from which came that RNA. Okay, it's a simple thing. So that was one of the methods that was used. The second thing was to create linkage maps. Again, linkage maps is something that I've already talked about in genetics. So we are going to figure out recombination frequencies whenever it's going to happen in meiosis. So in meiosis, during the prophase, meiosis 1, prophase 1, there is going to be recombination that is going to happen between the chromatids, and based on this we can figure out the linkage map, meaning that how one gene is linked to another gene, and based on this we can physically figure out where that gene is on a chromosome. So where exactly is it present, what is its physical location, that can be figured out, and that is something that I've already spoken about in detail, so refer back to that for this. So that is how linkage maps were created. And the third thing that they used was using BACs and YAC. BAC is bacterial artificial chromosome and yeast artificial chromosome. So these are plasmids okay. So bacterial artificial chromosome and yeast artificial chromosomes are plasmids, and these are artificially created, of course, not found in nature. And what is the use of these plasmids is that whenever you take a human genome, like I said, it's huge, it's really big, because it's going to contain somewhere around 3 billion base pairs. Now if you want to sequence all of the 3 billion base pairs one by one, it's going to take a long time. Also, it's not physically possible to sequence 3 billion base pairs from 1 to 3 billion in one go, and that is the reason why BACs and YACs are used to create something that we call genomic libraries. So whenever you take up the human genome in its entirety using restriction endonucleases, these are enzymes that are going to be cutting the DNA at specific places. So using one specific restriction endonuclease, you can cut up the entirety of the human genome into pieces, and each piece is going to be inserted into one BAC or one YAC. Okay, so each piece goes into one bacterial artificial chromosome. This can be put into a bacteria, and then it's going to replicate itself. So you will have multiple copies of BACs, and these can be sequenced. So sequencing is when you're going to figure out how they are arranged one next to each other, meaning each nucleotide, how is it arranged one next to each other, what is present after the first nucleotide. Let's say the first nucleotide was A, the second nucleotide was another A, the third nucleotide was a T, the fourth nucleotide was a C, fifth G, and then another G, another A. So you know the sequence of the DNA. So that sequencing can be done whenever a short fragment is being put into a bacterial artificial chromosome or a yeast artificial chromosome, and it's easier to sequence rather than taking the entire big 3 billion base pairs. So because you have cut them up, they are going to be anywhere between 100, 200, 300 base pairs to a few thousand base pairs, and these can be sequenced easier rather than taking the entirety of the 3 billion base pairs and sequencing them. And that is how BACs and YACs come into use, and these are known as genomic libraries, and then they are used for sequencing purposes. The fourth thing that happened was the whole genome was sequenced. So if you take each and every piece that is there in a back or a yak and sequence it, and each sequence is attached right next to another. So you had one piece, let's say this is the first piece, the second piece, the third piece, then the fourth piece. So you have different pieces of the DNA that is inserted into a single back. So this one goes into one back, this one goes into another, this one goes into another, this one goes into another. And these have been sequenced individually. And once they have been sequenced individually, you just lay the sequences right next to each other, and then you have the entirety of the sequence of the human genome sequenced. So the fourth and the final step was to sequence the entirety of the genome, which was done in this way. The Human Genome Project was an open source project. An open source project means that whatever the information that is gained from the project is available for everybody. Anybody can access it, anybody can use it, anybody can see it. So what are the applications of the Human Genome Project? Was that first it helped in understanding gene function, regulation and interaction. So because we sequenced them, and we also figured out that there is specific parts of the DNA that is going to give rise to an RNA. So there are functional parts and non-functional parts. So there are coding regions and non-coding regions. So when we did the Human Genome Project, it was figured out that close to 99% of the entire human genome is a non-coding part. There is only 1% of it which is coding for RNA. Any kind of RNA, not just mRNA, any kind of RNA that is going to have a function. So the coding region of the DNA is very, very small in comparison to the non-coding part of the DNA, and therefore we could figure out which part of the DNA is going to give rise to a functional gene, that is a coding part of the DNA. So we could figure out that here is a specific part of the DNA that is coding, and also if it is going to code for a mRNA, it is going to give rise to a specific protein, and that protein is conducting a specific function. So we could figure out that this part of the DNA has this function, and here is another part of the DNA that is going to give rise to a tRNA, and here is another part of the DNA that is going to give rise to a rRNA, which will take part in the ribosome as its structural part. Also, we could figure out gene regulation. Again, this goes back to operon concepts and operons in general, that how a gene is getting regulated. What are the other aspects that come into play for a gene to get activated or for it to get deactivated. And interaction, meaning that how the genes are interacting with each other. So if you again go back to the lac operon concept, you have the repressor gene, and that is going to synthesize the repressor, which is going to interact with the lac operon gene. Okay, so the repressor gene is different because it has a different promoter than the lac operon gene, which has a different promoter. So these are two different genes and they are interacting with each other. So we could figure out, or rather map out, how two different genes are interacting with each other. That was also something that we gained from the Human Genome Project. We could identify mutations because of the Human Genome Project. So because the entirety of the human genome has been mapped out and sequenced, we know that this particular gene needs to have this particular sequence. If there is an insertion or a deletion, or a part of the DNA is completely missing, or anything of that sort happens, meaning that there is a mutation, we can always cross-check it with the Human Genome Project that was already done. So that sequence of the DNA is already there, and any individual's DNA can be cross-checked with it, and we can figure out this part of the DNA is mutated in this individual. So identifying mutations became much more easier. We could predict diseases, which led to better diagnosis and better treatment. So again, diseases are caused by mutations at the end of the day. Any kind of disease is, at the end of the day, if it is genetic, is going to be caused because of a mutation. And if we can identify a mutation, we can already predict that this person is going to have a specific disorder or a specific disease because it has a mutation in this gene which is present on its chromosome. So going back to Mendelian genetics, we were talking about all of the genetic disorders that are there, and every single time there is a gene that is mutated which is present on a specific chromosome. Now all of this information that we have right now all came from the Human Genome Project. So we could figure out that there is a specific mutation, and only because we have the original to compare it with. And because we can predict diseases, we can diagnose it better. So we need not always wait until the person starts showing symptoms of the disease. Just by sequencing the DNA, we can already predict that this person is going to have this disease. And because we already know it beforehand, we can give better treatment and maybe stop the disease from occurring. Okay, so this was another help from the Human Genome Project. The last thing was to compare genomes of different species. So this is comparative evolutionary biology that we are talking about here. So based on the human genome, we can compare it to the genome of, let's say, apes. We can compare it to the genome of bacteria. We can compare it to the genome of mice. And we can figure out how a specific organism evolved, what is the evolutionary tree, by comparing the different genomes. So if we find parts of the DNA being similar to the part of the DNA of an ape or of a mice or of an owl or a deer or anything of that sort, we can figure out that this is how the evolution happened, because we have comparative genes between the two species. Another thing that can be done was to compare genomes within the same species but different populations. So if you take humans as examples, we have different populations. Like the population within India, again you can differentiate it as the population within the southern part of India to the population which is in the northern part of India, and we can compare the genes that are there in the people of northern and southern regions and figure out the differences that are there, and we can also figure out how these two populations evolved separately. Why are they different? How did they come about? So all of these things are applications of the Human Genome Project. Discovery from the Human Genome Project was tandem repeats and satellite DNA. Now what are tandem repeats? Tandem repeats are when one or more nucleotides are repeated over and over and over again, and they are placed one next to each other. So for example, let's take two nucleotides like A and C. Okay, so these are two nucleotides, and if they are repeated over and over again in this way and they are right next to each other, that part of the DNA is known as a tandem repeat. So it's one or more nucleotides. It need not be two, it can even be one. So you can just have A's which are repeating over and over and over again, or it can be two or three or many more. So one or more nucleotides which are repeated over and over again and they are placed one next to each other. So the AC is going to be repeated and it is right next to each other, and then it is known as a tandem repeat. Now tandem repeats are of different kinds. If two nucleotides are repeated like AC there, it's known as a dinucleotide repeat. If three nucleotides are repeated, then it is known as a trinucleotide repeat. Now if 10 to 60 nucleotides are repeated, then that is termed as a mini satellite. And satellite DNA are those which are present either in the centromere or the telomere region of a chromosome. So in the centromere region or in the telomere region of the chromosome is where you find the satellite DNA, which is nothing but tandem repeats but of a specific number. So if it is 10 to 60 nucleotides which are repeated, so 10 nucleotides or 60 nucleotides which are repeated and they are placed one next to each other, then it is known as a mini satellite. If it is 1 to 6 nucleotides which are repeated, or 1 to 6 nucleotides or more than 1 to 6 nucleotides which are repeated but less than 10 of course, and it is repeated from anywhere between 5 to 50 times, then it is known as a microsatellite. Now microsatellites are different from mini satellites because they have higher genetic diversity. So their genetic diversity is much more higher in a population. So if you take individuals within a population like a human population, if you take individuals within a human population, the microsatellite will have higher genetic diversity as compared to the mini satellite. Also it's going to have a higher mutation rate. So the mutation rate within the microsatellite is higher as opposed to that of the mini satellite, and that is the differences. One, it's going to be 10 to 60 nucleotides as opposed to 1 to 6 or more than 6 nucleotides, and this is repeated specifically 5 to 50 times, and this does not have a specific number for it. And the difference is that mini satellites does not have that much of genetic diversity, but micro does, and the mutation rate in case of microsatellites is much more higher as opposed to the mini satellite. Now, microsatellites are also known as short tandem repeats, or it's abbreviated as STRs. Now, coming back, the micro and mini satellites together are known as the variable number tandem repeat, or VNTRs for short. So micro and mini satellites together are termed as variable number tandem repeats, or VNTRs. Now, why tandem repeats and satellite DNA? Why are we studying this? The uses of it is that it is used in DNA profiling. Now, DNA profiling can be done in various fields. Majority of them are listed here. For cancer diagnosis, you can do DNA profiling. In cancer diagnosis, in kinship analysis, meaning pedigree, as in how your family tree looks like who is attached to whom, the genealogy of a clan or of a population if you're talking about kinship analysis. For paternity testing, that is self-explanatory. Forensic identification, this is when you would be doing DNA fingerprinting or things like that. And for genetic linkage analysis, that kind of gets into the kinship analysis, but it's pretty much the same. Genetic linkage analysis is wherein you're trying to figure out how genes are linked. And these are the uses. The majority of the use will be DNA profiling, and DNA profiling can occur in all of these different areas. So that is with tandem repeats and satellite DNA.